Assalamualaikum warahmatullahi wabarakatuh. Greetings to everyone. First, I would like to thank the organizer of the International Conference of Veterinary Medicine and Health Science for the great opportunity. I am Najma, an undergraduate student from the Faculty of Veterinary Medicine at Brawijaya University. On this occasion, I will present the result of my research on the development of a bioinformatic-based detection and building model of the MSPA gene in Mycolophobacterium fortuitum. Let's begin by looking at the background of this research. So, non-tuberculous mycobacteria, or NTM, generally refers to mycobacteria other than the Mycobacterium tuberculosis complex and Mycobacterium leprae. NTM can be found in the environment, such as soil and water. These bacteria can infect both animal and human. Many recent studies show an increase in NTM cases worldwide, caused by environmental changes, the unavailability of adequate detection tools, and a lack of awareness about the disease. The solution of this problem is to find an appropriate detection tool. One of the adequate detection tool is polymerase chain reaction, or PCR. So PCR is a detection method that amplifies DNA fragments at specific targets. To successfully amplify DNA, it is essential to have well-designed primer. Here we choose MSPA as the specific gene in Mycolophobacterium fortuitum. Mycolophobacterium fortuitum is a rapidly growing mycobacteria. The MSPA gene is a specific gene that encodes a nanopore protein for Mycolophobacterium fortuitum. This protein plays a role in the uptake pathway for hydrophilic nutrient molecules. MSPA is extremely stable against denaturation and capable for PCR. This study aims to develop primers and protein modeling for the MSPA gene in Mycolophobacterium fortuitum. Moving to the methods, first, the MSPA gene sequence was obtained from the NCBI database and used as a template for primer design. The FASTA sequence was entered into primer blasts, and then forward and reverse primer candidates were selected using primer blasts. Candidate primers were analyzed using NetPrimer, and protein modeling was performed using Swiss model. And here's the result. The FASTA sequence of the MSPA gene of Mycolophobacterium fortuitum shows that there are 648 nucleotides were used for primer design. After designing primers using primer blasts on the NCBI site, 10 primer candidates for the MSPA gene were obtained. This table lists 10 recommended primers from NCBI primer blasts. Each primer has a base length of 20 pp. All the primer pairs meet the base length criteria. This table shows the characteristic of 10 primer pair candidates obtained from NCBI primer plus. All primer pairs have ideal GC content, TM difference, and TM range. Primer pairs 1 and 10 have the least self-complementary values. Therefore, these primer pairs are suitable for primer candidates. This table shows the secondary structure of the primer candidates analyzed using NetPrimer. Yellow highlights indicate the primer characteristics that do not meet the parameters. The analysis results show that 6 primers do not meet the self-primer criteria. In the hairpin analysis, only primer 5 that doesn't meet the criteria. In the repeat analysis, only primer T3 that doesn't meet the criteria because it has 3 identical nucleotide sequences. The highest rated primer is primer pair 1 with forward and reverse primer rating of 100 each. This figure shows relative position of primer candidate, which is primer pair 1, towards MSPA family foreign gene. Primer 1 has an amplicon length of 148 pp, amplifying the region from 167 to 314 of the MSPA gene of Mycobacterium fortuitum. This figure shows the 3D protein model result of MSPA gene using Swiss model, visualized as an arrangement of alpha helix, beta sheets, and loops. This protein structure consists of 215 amino acids. This figure visualizes the hydrophobicity map with hydrophobicity values of minus 59.2. Hydrophobicity is crucial for protein stability and is a key in drug interactions with biological membranes. This figure visualizes the ionizability and the pKa value of 1990. The pKa value of ionizable groups impacts protein structure, stability, and functions. So based on the study, a specific primer pair, which is primer pair 1, was identified to detect the MSPA gene. This primer pair possesses the best quality and avoids the formation of hairpin, self-complementarities, and dimerization. The study also successfully developed a protein model from the MSPA gene consisting of 215 amino acids. The pKa value obtained was 1190, and the hydrophobicity value was minus 59.2. Here's the reference I used for this presentation. Okay, that brings me to the end of my presentation. I sincerely appreciate your attention. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh.